Hi, I'm Brian. You're going to see a lot of me today. It's the DIA day. I'm going to talk a lot. Sorry. Um, I figured I'd break this basically day up into a bunch of sections. The first section, this first hour, we're going to talk about some introduction to DIA, and then secondly, we'll talk about how to do it, and then particularly how to interpret data in Skyline. And then at the end, we're going to talk about how to infer data directly from, how to infer detections directly from DIA data using a program called Pecan that we've been working on. So to start, I owe a lot of thanks for this section, this first section, to Jarrett Eggertson. And this is an, no. Oh. All right. Oh my God. Perfect. Okay. I don't know why there's a light now above me, but perfect. So this this example is straight out of his playbook. So Jarrett was interested in the question of like going into a library and asking the question, where is Harry Potter in the library? And there's kind of like a couple of ways that you could think about doing this, and a fairly naive mass spectrometry way of doing something like this would be to pull a bunch of books off of the shelf and pull them out into a cart and then look through the cart and say, okay, I found Moby Dick, I found Pride and Prejudice, I found War and Peace, but I did not find any Harry Potter. And so then the question is Harry Potter in the library? Is still kind of outstanding, right? And a much better way of trying to ascertain whether it's there is just to go and look in the section that's appropriate based on the Dewey Decimal System and say that's exactly where Harry Potter ought to be. And therefore, because I see it or I don't, I know that it's either present or absent from the library. Now, this is sort of ridiculous. Obviously, with DDA, you wouldn't necessarily say that something wasn't present just because you didn't detect it. But there are a lot of times when we actually really need to know when something is absent. And I think that is a question that you can get at with sort of the targeted methods that we've been talking about, but also DIA. And I think that this is an important point. Failing to detect a peptide is not a proxy for its absence or abundance. And so we've talked a lot here about SRMs and PRMs, and they're great, but they're on a fairly limited pool of peptides. And so one of the questions that I think is on at least hopefully a few of your minds is how can we scale this up? How can we measure an entire genome worth of peptides? And is it possible to effectively target every single protein in a proteome? And the answer is possibly, but there are a bunch of compromises that go into that. And that's sort of what DIA is. It's a giant list of compromises. And so first we're going to talk about some of those compromises. We're going to talk about some of the approaches that you can use to acquiring DIA data, some caveats to acquiring it, and then when it's actually more beneficial to acquire DIA data than it would be, say, DDA data or PRM data. Okay, so let's first focus on those compromises. So with PRM, you're basically taking very narrow precursor isolation windows, these targeted windows, and measuring very specific peptides with fragmentation quantitation, right? And so when you're measuring fragment quants, it's stuff that has come through a Q1 and is mass filtered, fragmented, and then you're actually measuring it again, sort of like how you're doing with SRM. With DIA, you're doing the same kind of thing, but you're using sort of wide windows. DDA, on the other hand, acquires fragmentation spectra that are sort of like PRM. Again, they're narrow isolation windows. But instead of you targeting which peptides they're going to be, the instrument is actually going to target them. And because you're not getting regular scans throughout regular fragmentation scans, you have to do all of your quantitation at the precursor level. And so there are kind of like two ways to think about this. DDA is pretty comprehensive because you can use the instrument to target basically every peak that you, or not every peak, but many of the peaks that it can see in an MS1. But it's definitely not, the quantitation is all done at this precursor level. While with PRM you have pretty selective quantitation, but you have to choose some targets up front. DIA fits somewhere in between that. And the issue is dealing with these wide window peaks. Okay, and so really these compromises are about juggling some constraints. And there are two kind of main constraints. The first one is that you can only scan so fast without sacrificing your quantitative measurements, right? What does this mean? You need to be able to acquire a peak enough to draw, or you need to acquire enough points across the peak to be able to draw the shape of the peak. Okay, so that's constraint number one. And constraint number two is that to measure that peak, you need these 6 or 8 points across that peak. And so one of the issues with this is that the only way that we can get regular enough scans to get 6 or 8 points across the peak, across say a 30-second peak, is that we have to have fairly wide precursor isolation windows. And these wider windows allow a lot more interfering peptides because we're not fragmenting one peptide at a time, we're co-fragmenting a bunch of peptides simultaneously. Another compromise is that you are pretty limited in how long you can spend acquiring that peak. So with PRM, if you're interested in something that's really sensitive or down in the dirt, you can increase your dwell time and sit and park on that peptide for quite a bit longer and measure it at a higher resolution with higher time accumulating ions and get a much better signal as a result. And so the third way that you can limit this is you can also just say, I'm not going to try to actually measure all the peptides. I'm only going to try to measure peptides that have precursor charges that I expect or parent ions that I expect in terms of like the mass to charge range. Okay. And so we end up with data that's sort of like this. So this is a particular peptide that I was interested in. This is what it looks like if I measure it with PRM. This is what it looks like if I measure it with DIA. Actually, the signal is pretty clean if you consider the fact that, or comparable, I should say. So this is at about 10 or 10E4 basically, and this is at about 10E4, right? Or at E4, and this is at E4 basically. And you see the same signal there, but it's lower quality as in you've got fewer points across the peak, and it's also kind of sheltered by this interference. So we end up with data that's kind of like complicated to interpret, basically. So the first thing that I kind of wanted to go through is what types of DIA experiments people do out in the field and have done in the past, and then kind of hone in on a couple that I think are worth pursuing, or more modern ways of doing DIA. So just to give you kind of a framework, you've seen kind of stuff about parallel reaction monitoring, but I wanted to show you it in this sort of framework so that it makes the rest of the slides make sense. So with parallel reaction monitoring, typically you might acquire a precursor scan, usually just as a reference point, and then you'll acquire a bunch of fragment ion scans, say 4 in this duty cycle. And then you'll do another duty cycle where you acquire another precursor and another 4, and they will be exactly the same, right? They should line up one right on top of each other. And so because you're measuring these consistently, you can do that fragment quantitation that I mentioned earlier. Okay, so one of the first DIA methods that actually became reasonably popular was called MSE. And this was, I think, a pretty smart approach in where they would basically try to collect a precursor and then a fragment scan, and then a precursor and a fragment scan. And so basically they would acquire this precursor scan, you would see a bunch of peptides here, right? And then they would acquire a fragment scan, a fragment scan, and then that would contain every fragment ion that was associated with the peptides that were in that precursor scan. And it would span this same wide window. And the way they would talk about this is that there was a low energy scan, this precursor scan, and then a high energy scan where they were imparting enough energy to actually fragment the peptides. Okay, and so then they would alternate between high, low, high, low, high, low, right? And so here you would be able to get quantitative fragment ions. The only problem is that you're co-isolating all of the peptides simultaneously. So this was a method, this was one of the early methods. It had a lot of compromises, or emphasized these compromises pretty heavily. So as I said, the quantitation was done with fragment ions, but you do get fairly fast quantitative scans, usually less than a second for each of these scans. And so you get a lot of points across the peak, or it lets you be able to have very narrow peaks. And another advantage of this approach was that it did measure across the entire precursor range. It would measure from like 400 to 1600 or 200 to 1600. But the biggest issue here was that it had a huge reliance on computer algorithms to sort of deconvolve this data into its various peptides, because you were basically co-fragmenting 20, 30, up to 100 peptides simultaneously, and then asking the computer program to kind of figure out what was what. And so this data was like basically impossible to manually interpret if anyone has looked at it. Now this benefits substantially using sort of more modern techniques like ion mobility, and so they've modified this to be what they call HDMSE, but that's kind of like the way this framework works for MSE water style DIA. Okay, there was another approach that was released a little bit later called Pacific, which was out of the Goodlet lab. And the idea here was that they would acquire a precursor scan and then narrow fragmentation scans, kind of analogous to the width of what we were typically seeing with DDA, so that they would be 2MZ wide windows just tiled across. Now you can't get very far with 2MZ wide windows across a huge range. So what they would do is that they would acquire another precursor scan and then start over again. Okay? So is this DIA? It is, because it's in a DIA section. Is this, can you quantify using fragment ions using a system like this? And so are you going to get repeated scans from one duty cycle to the next? No, you're not. You're shaking your head. So consequently, you have to quantify using a system like this on precursors. But you do get pretty narrow fragment scans. And so this was the approach that was pioneered by the Goodlet lab, and they got really deep proteomes. But the quantitation, as I said, was all done at the precursor level. Okay, so so quantitation at the precursor level, fragment ions were only used for detection. It was just basically a way to guarantee that you would sample potentially every peptide at least once. And back then, they were actually using LTQs to do this type of analysis. So they ended up having to do like 50 injections to scan an entire range, because the speed of the instrument was pretty slow. It was only acquiring like 5 scans a second or something like that. Okay. But there was a huge advantage here, which was that these fragment scans you could actually just send into a database search engine like sequest and actually do peptide detection out of them. Okay. So the world has kind of been taken over by storm, I think, by this idea of what we're calling wide window DIA, which is, some people will go by the name of swath or just call it DIA in general. But the idea is trying to acquire an entire proteome using the same sort of framework that we were talking about with MSE, just sort of breaking up the fragment scans into precursor isolation windows. And so the idea was that you would acquire a precursor scan and then a narrow window fragment scan and then a second narrow window fragment scan and keep going and tile them across so that you can get repeated scans between each duty cycle. And so you would end up like basically in a situation where instead of co-fragmenting maybe 100 peptides at a time, you would maybe be co-fragmenting between like 5 and 10 peptides at a time. And so then interpreting these DIA data, while you couldn't just send them to a search engine, they were quite a bit more simple to interpret or to do detections out of than the MSE data, where you were doing high-low scans alternating one after another. So here, again, you would get quantitation using fragment ions. You get frequent enough scans to be able to do this quantitation. That said, you would end up measuring usually a kind of a compromised precursor range. So instead of measuring from 400 to 1600 like you would with MSE or with a normal DDA experiment, typically we would measure from 500 to 900 MZ, or from 400 to 1000, or 400 to 1200, but it's usually a pretty narrow scope. So a big advantage of this is that it's a lot less reliant on deconvolution. That said, it's still much too complicated just to give to Sequest or to Mascot to interpret. But it does start to look like PRMs. I keep hitting that button that turns it off. Okay, so what does a DIA experiment actually look like instead of in this toy example? So these modern DIA experiments don't acquire just 4 scans to measure an entire proteome over and over and over again. Typically, we end up measuring one precursor, again, say from 400 to 1000. We'll acquire a fragment ion scan somewhere in the range of 25 MZ wide, and we'll tile them one after another until we can cover the entire range. So here I've got 25 by 24 MZ wide windows. And then we'll acquire, again, another precursor scan. So to represent this a little bit further, I'm just going to take that and say these have about a 2.5 second between them, between the duty cycles. I'm just going to flatten this up, right, so that we can see this in a repeated way. So, so this basically represents one duty cycle. So if we take a look at one of those fragment scans, say this one from 784 to 808 MZ, and we look at what it looks like in a mass spectrometer, this actually represents a pretty high abundant peptide, but this is one scan that you might acquire. And you'll see the sort of normal B and Y ions that you would typically see with DDA. But the big advantage here is that because you're acquiring these repeated one after another, you end up with this sort of trace, right, where you see repeated scans one after another, and you can actually see the peptide rise and fall out of the background noise. And so because of that, you can draw these chromatograms that you've been seeing as we've been looking at with SRM data all week. Okay, so again, as I said, you get fragment ions that you would typically expect to see. This is what a DDA-like spectrum would look like for a single scan. And then basically we're trying to acquire repeated DIA scans and then look at them down this axis to see the chromatogram. All right. So there are some pretty big caveats that some of which we've started to touch on in terms of the wide precursor isolation windows. The first one is that you're going to see these peptides that come up in this precursor range. And one kind of important point to note is that the ions that you're seeing or the ions that you're measuring aren't necessarily in the middle of that isolation window. What that really means is that as a general result, the collision energies collected or for collecting these by the instrument aren't tuned specifically for that mass. They're going to be reasonably close because it's got to be somewhere in that window range, but typically the instrument will on the fly tune the collision energy for the center of that window and not for peptides at the beginning of that window or at the end of that window.
So the collision energy should be varying much more by charge state than by the slide mass.
Yeah. So what's the issue with that is, so that's absolutely true. One of the things that is a problem here, though, is we can really, we're acquiring these peptides and we're co-acquiring many peptides at the same time, right? And so charge state it becomes a problem because we have to make an assumption about what the charge state of our peptide is, right? Is it a +2 or a +3? Well, there's going to be +2 peptides in there, and there's going to be +3 peptides in that same precursor isolation window. And so not only are we tuning the collision energy based on the center of the window, but we're also saying, I'm assuming all peptides are charged 2, right? And so your charge 3 peptides, your charge 4 peptides are going to have very different collision energy tunings that are associated with them. And so, so consequently, there are multiple peptides in this range, and you might have one that's a +2, one that's a +3, one that's a +4. And they're all fragmented the same as if you're assuming they're +2 peptides. Okay. One additional sort of issue with this is that there are these edge effects that can affect the signal transmission. What does that mean? Okay, so usually quads aren't perfect, right? So quads transmit ions of basically a square wave, and we assume that that any ion from 784 to 808 is transmitted equally in equal proportion, right? But realistically, I mean, it's a device like any other device, and it's got tolerances, and so typically quads tend to transmit not so perfectly. They have some rounding at the edges. And so one way to sort of deal with that strategy is to effectively widen the windows here. But that's definitely something that's important to know, that depending on the make of your instrument, you might have different sort of transmission efficiency problems depending on the instrument type. We'll talk about that a little bit later. Okay. So looking at what this data sort of looks like, this is a trace across a 90-minute gradient for this particular superoxide dismutase peptide. And so these are all the fragment ions that are associated with superoxide dismutase, this one peptide. And so if I were to ask you, like, where is this peptide, it would be kind of hard to pick it out from here. But that's basically where the peptide is. And if we look at the trace of it, it has kind of a nice shape. It looks like a reasonable peptide with lots of different fragment ions that we can call. And it's relatively easy. It stands out from the baseline here. Okay? So this is kind of an example of what good DIA data looks like. It basically looks like good PRM data. But here is another peptide from exactly that same protein, superoxide dismutase. And this one's a little bit harder. So I'm going to zoom into this one region over here. And this is the trace, right? And so it looks like there's a peptide here or a peptide here. I want a show of hands. What do you guys think? Do you think it's this peptide over here is the indication for it? It's kind of hard to see on these screens, but if you've got them up, you can pick them apart a bit. Or over here. This one? This one? It's that one. So how can I make that call? I have to use a computer program to make that call. But it's kind of amazing to think that this data is as complex as we're looking at. And you actually do see the co-occurrence of many of these fragment ions to the wrong retention time. We know that at least one of these is wrong. This peptide isn't eluting multiple times, right? So what's going on with that particular peptide? What does that actually look like? So if I look at a library spectrum, so this is a spectrum for that specific peptide here, I get a bunch of B and Y ions. And if I look in that sample, I can see a bunch of the same patterns here. I can see that it's got pretty high Y8, Y9. It's got relatively low everything else. Maybe the best ions that I can pick out are this Y10, Y11, Y12, right? And you can see that those are the highest ions in this trace as well. And so that's actually how the computer program is trying to figure this out. And one of the things that's actually kind of intriguing, I think, in this experiment is, you see this in the pure sample in your library. You've got, a much higher, or a little bit higher Y8 than a Y9, but we see the higher Y9 in our acquired data. What do you think is going on here? Yeah?
Could this be related to the charge collision energy issue you mentioned?
Yeah, definitely could be. So here, this is a +3 peptide, so it's fragmented with the wrong fragmentation patterns. So, or the wrong charge state assumption, basically. That's definitely one potential concern. What are other ways that you could actually get peaks that are out of place? Yeah, and that's actually what's going on right here. So if I look at, oh my.
Repeat what you said.
Yeah, it could be another peptide. And so what's going on is if I actually highlight the ions that fit this peak shape, it's actually pretty easy to pick out. Yeah, that actually looks like a peptide, right? You get enough co-eluting ions that evolve, they rise and fall together. But there are a bunch of these ions, so the ones in yellow maybe are useful for detection, but maybe aren't so good for quantitation. And that's what that's what the Y9 ion is. It's this one that's like a little bit higher than everything else because there's something else that's co-eluting with it. You also get a lot of these traces, these red traces that really are sort of representative of the background signal. These might just be from other peptides, or they're definitely from other peptides, but it's clear that they are not rising and falling necessarily with the shape of this peak. And a bunch of these particularly low MZ ions aren't necessarily reliable in terms of being able to make a call on them because they tend to co-occur with other peptides pretty frequently. Okay, so that's what's going on in this peptide here. So I've got an example here, and this is kind of an interesting peptide. And I wanted to ask, what's going on here? We have to make the assumption that peptides don't elute multiple times, right? Otherwise, I think you're kind of like totally screwed with DIA. We have to assume that every peptide is going to come off the column once. But we see kind of like this nice signal in two different places, right? For the fragment ions, these are the precursor ions. Again, this is pulled straight from Skyline. So the fragment ions actually look like they agree exactly, right? In terms of you get the green one, and then the yellow one, and then this blue, and then the purple one, and it's the same ordering here too, right? So they look like exactly the same peptide. One of the things you see is that the precursor trace is a little bit different. And this blue ion here represents the monoisotopic peak. And so this one has no monoisotopic peak. It only has a +1, +2, and +3 fragment ion. So this is actually a deamidated form of the same peptide, right? So deamidated peptides are going to fall within the same precursor window because they're only shifted by 1 AMU, right? So if it's a +2 peptide, they're only going to be mass shifted by half a Dalton, half an MZ, and so they will definitely fall in the same precursor isolation window. And so we'll collect them one after another and then have to make the call, is this the peptide we're interested in or is this the peptide we're interested in? We'll talk a little bit about these kind of concerns in a bit. But I think this is kind of an important caveat to bring up now. Here's another example. So this is actually a phosphopeptide. This is a phosphopeptide from mTOR. So we've got a really nice signal here and a really nice signal here. Which one is the peptide? Well, I know that this one is the peptide, but what's that other signal from? That's exactly right. So the idea here is it's the same peptide. It's got the same number of phospho sites on it, basically. It's been phosphorylated once in this case. But this one is phosphorylated at the canonical known site. And this is the same peptide, just phosphorylated at a slightly different site. That's going to change some of the fragment ions. It's going to change when it elutes, but the precursor mass is going to be exactly the same. Yeah?
Is there a possibility of in-source fragmentation giving you the same? So in-source fragmentation, your phosphorylated sites, for example.
That's probably not going to be the case for phosphopeptides because what does the phosphate lose with it? It loses water as well, right? So you don't get a neutral loss of 80, you get a neutral loss of 96. And so it will appear as if it's 16 down in its precursor. But say we have a precursor isolation window of 24 MZ wide windows. In that case, you might actually see it at the same time in the same place.
The caveat I was thinking, when you're running like longer batch and your calibration is going up, would that affect your, the percent CV, for example? The reproducibility in your, like the resolution is going up.
Yeah, so definitely like resolution is for example, I think what your question is, if the calibration changes over time, do I end up having peaks come out of the window, of the precursor isolation window?
I'm saying the reproducibility of those things, the quantitation.
Oh, yeah, yes. So definitely low. So can a peak basically like have so much error in it that you actually aren't measuring it anymore? That actually definitely is a problem here sometimes. Actually, it's a much bigger problem in orbit traps than it is, I think, in tops. For a couple of reasons, but I think the primary one is, so it would make sense that your low abundance ions are going to have less mass accuracy, right? Because you have less points to figure out what the actual mass of that peak is, right? And so low abundance ions, you're going to have, potentially, those are the ones that are going to get shifted out first. That's what you would assume, right? In an orbit trap, actually, you end up with an alternate effect on the top end as well, as a result of coalescence. That's where if you've got a high population of one type of ion, say like 90% of the ions that are in the trap are of one specific type, which definitely happens if you've got one specific peptide, for example, you can actually, you end up, the orbit trap ends up like drawing the masses of the other fragment ions towards that mass. And so it basically like will pull them off of whatever mass accuracy they were supposed to hit. So in those cases, you actually get a higher error at the top end than you do when you've got like one big fragment ion at the top end than you do otherwise. Does that sort of make sense? Yeah, so you do see things that will sometimes pull in and out of the window. That said, this is actually less of a concern for DIA, I think. Those issues are less of a concern for DIA than they are for DDA. I think you're much more likely to see the fragment ions that you're interested in with DIA, and you don't have quite as much of an effect of these mass accuracy issues. The reason why is because, like coalescence, with coalescence with DDA, you just need one peak somewhere to pull everyone off of track. And so in your precursors, for example, when you're doing your precursor scan, if you've got just one really abundant peptide, say you've got like a big keratin or something like that, or a big trypsin peak, it's going to shift the precursor masses of all of the other peptides away from what their true mass is supposed to be. With DIA, because we're doing these these wide window, but much narrower window than 400 to 1600, you actually have a much less chance of seeing another big peak along with your peptide of interest. Does that sort of make sense? Do you guys kind of get that? It's definitely an advanced topic. I can leave that for the advanced section next. But yeah, does that sort of answer your question? Okay. So we can get peptides that have the same or similar precursor masses that fit in the same precursor isolation window that share a bunch of fragment ions. And that complicates our ability to do the analysis. So basically, I've given you like a bunch of ways in which DIA kind of sucks. But there's got to be like some reason to do it, right? So why would I use DIA if there are all of these different caveats for problems that you can get with DIA? Okay, and the biggest reason to do DIA, and I think the reason why people are interested in here in this room as well as in the community at large, is SRMs are really hard to do. SRMs and PRMs are hard to do correctly. And the reason why is because first thing you got to do is figure out what you want to target. You have to say, I actually have a hypothesis about my biological system. I can't just do a fishing experiment. I need to know, I want to measure metabolism proteins, and not just metabolism proteins, but these specific peptides from those metabolism proteins. So you have to generate a bunch of hypotheses about what you're interested, what proteins you want to talk about, and then you have to pick which peptides out of those proteins you want to go after. Then you've got to measure those peptides independently in a pure state or something where you have an idea as to when they might elute, for example, and figure out some retention time scheduling. And then you've got to pick which transitions you want to actually measure using your triple quad. This is less of an issue with PRMs, but you still need to figure out what transitions you want to annotate afterwards. And then finally you can get to this data acquisition. And probably you made a mistake somewhere in the line, right? You picked the wrong peptide. This peptide isn't measurable, or doesn't give you a good signal, or doesn't have good CVs, or it isn't in the linear range, or many other reasons why this might not be a reliable peptide. And then you have to go back and maybe select another peptide for this protein, and then figure out some new scheduling and do it again until you actually build an assay. And once you've actually got your pool of peptides, then you can verify you could test your hypothesis, decide if you were right, and then realize, oh, maybe I should have measured a different protein instead. And then you got to go all the way back to the beginning, select some new peptides, and then keep going all the way back down. This is a really time-consuming process. It takes a lot of effort to build an SRM assay from scratch. And I think that's just like a fundamental limitation of what we are dealing with targeted proteomics. DIA makes it such that we can go in without a hypothesis. We can do that fishing experiment and actually come out with something in the end. So the beauty, I think, of DIA is that it basically inverts the problem. It lets you acquire data before you've picked what proteins you're actually interested in measuring. Then at that point, once you've acquired data, you can say, I'm interested in measuring these particular proteins. Let's go extract them out of our library. So you pick a bunch of these peptides, you'll extract them out of the library, you'll do transition refinement in the same kind of way, and then you will say, did this make sense? Did the biology actually pan out? Do I'm seeing a disturbance in the mitochondrial pathway? And then you'll say, maybe it didn't. And then you can actually go back and say, okay, well, what if something else happened? What if there's some change in oxidative stress? Then you can go back and generate a new hypothesis, pick a bunch of new peptides, and just do this on the same data, right? You can do this at your computer instead of having to reacquire this data over and over and over and over again for every new hypothesis. I think this is the strongest beauty of doing a DIA method, and the biggest reason why we would want to. It's much less about being able to measure the entire proteome. It's about having the ability to not have to think about what you're doing while you're doing it quite as much. You can collect some data, you can experiment and play with it and iterate very rapidly on the data file instead of with the instrument itself. Okay, so that's reason one why you might want to do this. Reason two is if you're already collecting DDA data, you already have a mechanism to do basically that same thing, which is to acquire an entire proteome's worth of data and ask questions of it one after another as you've done. So I think one of the best things that we've seen out of DIA is that it is substantially more reproducible in terms of the measurements that you make. So for example, this is a DDA experiment I ran in triplicate. I would typically see around 35,000 peptides out of this HeLa lysate. And if I were to measure in triplicate, I only get about 60% coverage where all of the experiments agree. Now, sort of why is that? What's going on here? That's definitely one concern. False discovery is definitely one concern. I think the biggest thing is that for a lot of the low abundant ions, you're basically telling the instrument to pick what it thinks it wants to measure at any given time, right? And it's going to make a different decision every time because it has no knowledge of the previous acquisition. It has no knowledge of anything that's happened before. It's just making that decision on the fly. Should I measure this particular peptide? And for the low abundance stuff, the proteome is vast enough that there are bunches of peptides down at that lower range. And today you might sample one of them, and tomorrow you might sample a different one. And so the whole set of this is actually probably more representative of what the proteome is than any one of these individual scans. With DIA, we end up acquiring the same precursor isolation windows one after another every time. The instrument doesn't make any choices. We've made all the choices up front. Because of that, because we're doing this regular scanning across the proteome, we end up getting a much more sort of consistent measurement. So not only are we talking about the possibility of detecting more peptides, in this case like around twice as many peptides, we're also talking about the ability of being able to reproducibly measure every one of them, right? Or much more of them than you were typically doing with DDA. So this, I think, is the other big advantage of DIA. So not only is it better than PRM or SRM because you can do all of your decisions after you've acquired the data, but it's better than DDA because it has this big reproducibility advantage. And this manifests itself not just in terms of the detection level, but also in terms of your quantitative accuracy. So this is a data set acquired by Lindsay Pino, who has spoken basically the most of any of us, I think, this week, because she's a great scientist. And so this data set shows coefficient of variation at the bottom for all of the measurements for all of these different peptides. And basically what she sees is if she sees repeated signal across her measurements, as in she gets at least 3 transitions and at least 2 peptides for that protein, these peptides are remarkably reliable. And so if you're looking at like a typical 20% coefficient of variation that you expect, she's actually seeing about 90% of these peptides fall within this tight CV range. And this is extremely powerful, I think, quantitatively. This doesn't speak necessarily about the accuracy of the quantitation, and I think that's an important caveat, but it does speak about the precision of the quantitation. I think Lindsay at the very end of today will talk to you about how accuracy actually can be measured in these kind of frameworks. So again, with her data set, she sees this like beautiful 8% median CV for her peptides. And this is fairly typical, actually, of these sort of more simple experiments. If you're working in yeast or E. coli or a smaller model organism, this is typically what we end up seeing. If you're working in human cells or tissues, the CV is larger, not just because of the proteome, but because of the complexity of actually doing the sample prep. But the idea is still very much the same in terms of we typically get much lower CVs than you do with a DDA experiment. Okay, so just to kind of like, yeah.
Previous slide, I'm assuming these are 3 injections. I might have mistakes. 3 injections of same sample.
3 injections of the same sample. Yeah. So it's not even, yeah, we're not talking about like 6 injections or 10 injections or 20 injections. This is just 3 injections, like a normal triple kit. That said, DIA actually does enable us to be able to do high-N replicates, which I think is the beauty of DIA as compared to DDA. So with DDA, you end up, if you acquire like 10 replicates of a sample, you end up sampling different parts of the proteome one day versus the next. That said, a 10 injection pool is really hard to do with TMT or something like that. Something else that's another sort of strategy for doing quantitation. If you wanted to do 10 replicates of 2 samples, that's a much harder experiment. Really, with TMT, you're kind of limited to the 10 samples that you can measure, the 8 samples you can measure with TMT or eye track. And so DIA, I think, makes it much easier for us to be able to do large experiments that are spanning across weeks or months of acquisition, potentially. Okay. So just to kind of finish up on this first section, then we're going to do like a little coffee break, and then we'll come back and then talk, okay? So DIA is all about compromises. It's trying to do a DDA-like experiment, but using sort of the ethos of a PRM experiment. That there are many different ways of doing this type of acquisition. We talked about MSE, we talked about Pacific, we talked about swath. And we're going to talk about like a couple of optimizations on the sort of swath or sort of modern DIA approach in the next section. But there have been historically a bunch of different ways of doing this type of data. There are a lot of caveats that you have to consider. The one is that collision energies are not necessarily optimized, that you end up acquiring multiple peptides simultaneously, and that you have these edge effects. But I think that we see that the benefits of DIA actually really outweigh the sort of negative consequences of doing DIA, particularly in terms of the reproducibility of the experiments and also the ability to do this iterative refinement. Okay, so we have a few minutes. Do you guys have questions about sort of this early stuff? How many of you guys have been doing DIA already? A handful? Okay. And how many of you want to be doing DIA? Oh, okay, that's way more than any other class. That's amazing. Okay, great.
I have a quick question. Forgive me my ignorance, but I'm pretty sure that there are a lot of benchmark experiments already published, but I haven't read up on them. So how does DIA perform in comparison to DDA and TMT and time methylation and all the other techniques when you do spike-in experiments? So I mean, reproducibility, of course, is very, very nice, and if you can show reproducibility in replicates, it's also very, very nice. But I mean, at the end of the day, you want to see differences in abundance. Yeah.
So I think I have some slides that show DIA versus DDA for precursor ion integration, and I can make sure that I've got that for the next section. But we've done that sort of comparison, label-free versus label-free, and those typically we get much better results with DIA than with DDA. With labeled experiments, you can get better results. SILAC, I think, is sort of fundamentally limited in some ways because you basically doubled the precursor space, basically. For every one of your peptides, you now have 2 precursor signals, and so it's much harder to, that means that you get, every peptide you get 2 chances at being able to detect it, but at the same time, you also have so much more that the instrument is kind of overwhelmed. And so it's much harder for it to actually sample everything. iTrack, I think, and TMT is maybe the sort of most reasonable next method. That only works with certain caveats. You have to be doing MS3 acquisition. So does that make sense? Like MS2 fragmentation with iTrack actually gives you pretty wonky quantitation because you guys know how iTrack works? Okay, so you label all your peptides with a bunch of labels. And so the same kind of problem that we have here actually is a problem for iTrack. So when you acquire an iTrack peptide, you'll do a precursor isolation window around that peptide, 2 MZ-ish or whatever. And there's going to be that one peptide, and it's going to produce reporter ions. But there's going to be a bunch of other peptides that are in there. Some of them are going to be high abundant, and they're going to like skew in one direction. But in that 2 MZ wide space, I guarantee you that there are hundreds of low abundant peptides also that are just below your threshold, okay? So all of those low abundant thresholds all produce the same reporter ions. And so consequently, you get what we think of as saturation or sort of interference, but really it's like all those low abundant peptides are producing the same reporter ions, and they all average out to 1 to 1 to 1 to 1 to 1, which is why we get compression with iTrack. And that's why you need to do MS3 to actually pick specific fragment ions to be able to go after that peptide specifically. So that requires that you, A, have an instrument to be able to do that, so you're limited to a Lumos, basically, and to be able to do that fast enough, basically. It basically is pretty limited in terms of the amount of measurements that you can make in any given experiment. Consequently, like one of the things you have to do with that experiment is do pretty heavy fractionation, chemical fractionation, like solid, like a resin-based fractionation or something like that. So you have to do SEX or MCX or something like that, high pH reverse phase, to collect 10, 12, 24 fractions and then run each of those individually. So you lose basically like the biggest advantage, I think, of iTrack, which is the idea that you can multiplex a bunch. Now you have to do a bunch of fractionation and actually do the acquisition. So, but that said, I think that's the only thing that actually will achieve the same level of precision as you would get with something like DIA. And so some people have gone back and forth as to whether iTrack or TMT is better than DIA. There was a presentation at ASMS by Lucas Reiter about this idea, about comparing the two. But I think that's sort of like what's a reasonable landscape. Does that seem like a fair representation? Okay. Yeah.
What's the impact of having or not having the right spectral library for peptides and DNA?
That's a big problem. Yeah. So it's important to have a good spectrum library to be able to do acquisition. And in the second session, we're actually going to go through some examples and show you what that data looks like and what it looks like to analyze it naively without a spectrum library. That said, in the very afternoon session, I'm going to show you a strategy, and you're going to get a program to take home with you that allows you to do peptide extraction as if you're doing a database search with this program called Pecan. And so that'll be a take home that you can show to all your friends. It'll be great.
What's the difference if you don't have, suppose you don't have the spectral library, but you're using IRT, that will be a reduction time of your peptide? So like, that, I mean, can you trust IRT for that?
I mean, that I think will make it better than going in blind. But I would argue that if you've got an IRT, that means that someone has acquired that peptide, right? So you would be better off using something as a library. Even if it's something that someone else acquired and measured that IRT, you might as well use their fragmentation patterns as well.
No, I'm using non-peptide to predict the retention time of unknown peptide. I mean, which I don't know where it's eluding.
Those prediction programs are garbage. Like SSR Calc and that sort of thing. Like it's enabled in Skyline, but they're really not worth using as a starting point for DIA analysis. They're pretty dangerous in that regard. Unless you've got something that's like a special cooked up by yourself kind of thing that you've got. I would be pretty nervous about using something like SSR Calc to pick peptides or to pick where peptides should elute. Because that will be that peptide range, but plus or minus 10, 20 minutes. Actually, the biggest weight for SSR Calc is the precursor mass. So you could do just as good as if you lined up with precursor mass, basically pretty close to SSR Calc 3.
I think that's true, actually. I mean, like in our hands, like prediction of the SSR Calc in our gradient, like was plus minus a couple minutes. Like it was actually pretty accurate. Like I think it.
How long was your gradient?
25 minutes or so.
Okay, so yes, yeah. I'm talking about plus or minus 10% of the gradient.
It's still like it makes a difference, right? Like if you are searching for a peptide during the entire gradient round, it can be 25 minutes, or if you have to do it in 5 minutes, it's like it increases your chances to pick the right one by fivefold at least.
Actually, I would argue that it doesn't. And the reason why is because precursor mass actually, so retention time scales pretty linearly with precursor mass. And so if you know the mass of the peptide, which you do because you've got this isolation window, you can pretty well pick out where it will have eluded to a reasonable degree. What we find out with doing pecan analysis, that's going in blind without any kind of knowledge of what the peptide fragmentation is or retention time is, if we include SSR calc as one of the features, it doesn't perform any better. And that's kind of how we've come to that conclusion, that SSR calc is a good starting spot, but it's not something you would want to necessarily trust to give you a leg up with DIA.